Podcast, episode 41. Today I'm joined by Dr. Brad Shaffer, who's a distinguished professor in the Department of Evolutionary Biology and the Institute of Environment and Sustainability at the University of California, Los Angeles. Dr. Shaffer has published over 300 peer-reviewed publications ranging on topics from invasive species ecology to different aspects of quantitative genetics and everything in between. He's also done a lot of work with turtles and turtle phylogenetics and relationships. We're really excited to talk to him today. So thanks for coming on.
Be here.
Ken, do you want to start us off with the first question?
All right. This is the first question that we ask pretty much everybody that comes on our podcast. How did you get interested in turtles?
How did I get interested in turtles?
Yeah.
Well, so I've been interested in turtles since, I don't know, probably I was 4, something like that. So I always think there's kind of 2 types of people. There's people who took a class in college or something, and that sort of changed their life. And there's other people who just, for whatever crazy reason, just became sort of mildly obsessed with a group of animals when they were a little kid, and I was that latter case. So I was raised in Connecticut, although I've spent the vast majority of my life in California. And I was raised in a little town outside of New Haven, Connecticut. We had a pond. That pond had some snapping turtles in it. I loved them. I, caught them and played with them and raised them and did all kinds of stuff. And that's what got me interested. And then that took me through all kinds of different aspects of turtle biology and learning about turtles and where the world of turtle systematics and stuff was at. As I was in high school, I just sort of developed that interest on my own. So that's where I started.
That's interesting. That was something I was sort of curious about, because for me, it's sort of, I wouldn't inherently be interested in the genetics aspect and the classification aspect of things, and the more the realm of things you can't see, the coding of the organism. And it's something that for me was born out of looking at pond turtles and sliders and thinking that there's something to this that's more than you see at the surface. I'm curious if your interest in genetics and evolutionary biology was something that came from your interest in the animals, or was that a separate interest that developed?
Yeah. Well, it was more or less a separate interest, but so the history there is that, or the pathway is that, most little kids aren't interested in genetics. They don't even know what it is. And I certainly was one of those little kids. We had no science in our family. My parents didn't go to college, so there wasn't a deep history of any of that kind of more academic stuff going on. A lot of respect for education, but not a lot of history with it. And for me, I was always interested in evolutionary biology. I was always interested in the process of speciation. And how the process of speciation plays out then to generate closely related and less closely related sets of species. That's phylogeny in many ways. I always just found that to be an interesting puzzle and something that I cared about a lot. And when I was an undergraduate, so that was in the 1970s, the early and mid 70s, that's when genetics was just starting to be applied to natural populations to learn about everything from sort of classical population genetics to aspects of evolutionary biology, phylogeny, species limitation. And so that was in the old days of Alzheimer's electrophoresis. That was when it was just getting started. People were just starting to travel with liquid nitrogen tanks to get tissue samples. And as an undergraduate, first at UC Santa Barbara and then at UC Berkeley, I was exposed to that, and I just thought, this is really cool. And really again, not because I was enamored with genetics, but because in my mind, it just opened up windows and doors and opportunities to learn about the ecology and natural history and biology of reptiles and amphibians and in ways that, it just couldn't before. I mean, doing long-term market capture studies is a very, very difficult, time-consuming thing to do, especially with long-lived organisms like turtles. But you can learn an awful lot about movement patterns by studying population genetics, and that just really, spoke to me. So I kind of came at it from that point of view. I mean, the focus was, it still is for me, has always been, I want to learn more about the animals.
Yeah.
I like the animals. I love the animals. I want to conserve the animals. And I have always viewed the genetic tools that were available as tools that would let me do that in a more complete way and a more satisfying way.
That's really interesting, and it's a very different approach. There's sort of a physical and the reality you can see, but then there's that microscopic level of detail that it's not everyone's area of interest, but that you found that is.
Yeah, but if you think about, say, the area of molecular ecology, well, the ecology is what's at the center of that, or for most people. Most people don't really care about the molecular side of it too much. How you score a microsatellite, or why some genetic variant, came into being or exists, or, what difference it makes to the organisms in many ways. What they care about is the ecology. They want to understand the population size. They want to understand the history of population increases or decreases. They want to understand who mates with who. They want to understand if that clutch of turtle eggs had more than one father. Those are the things they want to understand. And the molecular, I mean, that's the window. That's the opportunity that lets you figure that stuff out. And that's what's cool. I mean, for most of us, that's what's cool about it.
Right, the code, the tools.
Yeah,
That's the tools
it's a tool
to
right,
figure out a
yeah.
story.
And to figure out the ecology that you'll really understand.
Right.
Yeah.
So I think we can sort of jump right into this, and that's kind of the fascinating landscape we want to discuss with you. There's something about turtles just right away that I think it's good to discuss. This is an area for a lot of people that is somewhat foreign. But turtles typically, when you're analyzing genetic data, it's all about the rates of change of single nucleotides, and that's what sort of at the gene level and the genome level changes. But with turtles, the rate of that evolution is slower than in most vertebrates, tetrapods, or amniotes. And I'm sort of curious. That's a fact, but why? Why is that something that's
Why does so are you asking why is that the case for turtles, or are you asking what are the repercussions of that for studying turtles?
I would say both of those things. Yes. Yeah.
So why is it the case? I mean, we don't know for sure, but it's linked to generation times. So turtles have long lifespans. They also tend to have long times to long generation times, right? So the amount of time it takes for a turtle to go from an egg to being a sexually mature adult is very long.
Right.
I mean, if you think about it, it's on the same order of magnitude as humans, right? It's usually about a dozen years or so is about the time it takes. For a green sea turtle, it's 30 years. For a painted turtle, it might be 5 or 6, but it's pretty long as these things go for vertebrates. And that evolution occurs by accumulating mutations and then passing those on to your offspring, and recombination and other things that happen that we all remember, hopefully, from our introductory genetics classes. And those basically happen once per generation. So if you are thinking about the rate of change at the genetic level in years, well, if you pack 8 generations, 8 opportunities for change into a century, it means you're going to get less change than if you pack 100, which is what a house mouse does, or 200 into a century, right? And it's just the generation time is long. That means the rate of change is slow, and that's it's true for long-lived organisms with long generation times in general. There may be something more going on with turtles. They have relatively low metabolic rates, and there's some connections between your basal metabolic rate and the rate of molecular evolution. So they're ectotherms. A lot of them hibernate, a lot of them really slow down metabolically, and that has some repercussions. So that's, as best as we understand it, or at least as best as I understand it, those are the sort of basic reasons why it's slow. Now, the repercussions of that for studying turtles, just remember that for almost anything where you're using DNA sequences to learn about organisms, it's all about, similarities and differences, right? So just to take an example, if you've got a clutch of eggs of a marine turtle or a painted turtle or whatever turtle you like, you've got a pile of eggs. A question you might be really interested in is, presumably one female is the mom of all of those eggs. What you don't know is whether one male is the father of all those eggs. It could be they mated, and maybe his sperm, fertilized all of them, or maybe she mated with a couple of males. And so in that clutch of eggs, there are full sibs that have the same mother and father,
Right.
and there are half sibs that have the same mom but different fathers. And those are quite different. There's all kinds of reasons why one might be curious about that. Well, how do you figure that out? Basically, at the sort of simplest level, you sequence a bunch of DNA from each one of those hatchlings, and you say, is there, a pile of them that are all genetically quite similar,
Right.
because they have the same mother and father, and another pile that are similar to themselves but different from the first pile, and that's because they have different fathers, okay? Although they have the same mom, and that more or less that's how you figure that out, okay? Well, if the rate of accumulation of mutations is really slow, then you don't have as much information to identify those differences. Right? Because you just haven't, so if there were two fathers, but those two fathers were themselves, shared a common ancestor a few generations ago, if the rate of mutation is really slow, then you're not going to be able to distinguish them. And so you're going to miss it. And so basically it sets limitations on being able to use genetic data to learn about very recent things in the recent past.
Yeah.
So similarly, you might, if you're a conservation biologist, be interested in whether a road is a barrier to movement of turtles on either side of the road. Okay, well, that road went in 50 years ago or 100 years ago or whenever it went in. Okay. So what do you have to do? You kind of have to see differences across that road that have accumulated over that amount of time. And, if the road went in 50 years ago, some of the same turtles that were there before the road went in may still be living there. And so, they're telling you about what happened before the road based on their genetics. And whereas if those were fruit flies, and every 90 days the population turned over, there'd be lots and lots and lots of opportunity for differences.
Yeah, right.
Right. Whereas with a long-lived animal with long generation times like a turtle, it just takes time to accumulate things because the rate of evolution is slow. So it makes it much harder to learn about things that happened in calendar years recently, right? Because it might just be one generation.
Yeah, right.
Yeah.
You've just got less data in a set period of time.
You've got less time for differences to accumulate.
Right.
And in many, many cases, differences versus similarities is the key thing that you're using to learn about them.
Right.
Right. And you just haven't had enough time for those to accumulate. Now, we can make up for that nowadays to some extent. because, we can sequence the whole genome of the turtle.
Yeah.
And that means, the average turtle has, as you said, Michael, that, we focus on these nucleotides, those are the building blocks of your chromosomes and your DNA. And the average turtle has about 2.5 billion unique nucleotides in its genome.
Comparable to humans.
So yeah, I mean, it's most turtles are a little bit bigger than humans. But they're very comparable. And so that means that any given nucleotide may not have much information in it because of time and slow mutation rates, but there's a lot of them.
Yeah.
2.5 billion is a lot of opportunities. And so if you can sequence, and one of the most important and exciting directions that have emerged in using genetics to study evolution and to study ecology is that it's getting easier and easier and easier to sequence the entire genome and sequence it well so that there's very few errors. And that just gives you, it gives you a lot of information. It still doesn't always make up for that generation time effect, but it helps because there's just so many more opportunities for you to see a variant that you can work with.
And you got a really interesting perspective with that. I mean, it's fascinating, but you've worked with this over the course of your career with turtle phylogenies and relationships. And so not only have you seen the phylogenies themselves change, but you've seen the types of analyses change.
Right.
And so I think we'll definitely touch on the genomics aspect, but maybe taking a step back into earlier attempts and some of your earlier attempts at constructing these phylogenies. There's something kind of interesting. There's so much, like you said, sort of early on, there's not much in science that is more holistic than the phylogenetic tree in terms of all the pieces of data and ways to interpret it. It's just as much sort of a philosophical construct as it is a computer science question. So it's kind of a fascinating, it's all these different angles you can take. But early on, before you had all this genomic technology and the things that are just becoming available now that you're sort of at the forefront of, what was the way that you would work on these problems? And what were some of the common issues you had, for instance, using just single genes for relationships?
Well, I mean, in anything, right? I mean, whether it's genomics or putting radio transmitters on organisms or, whatever it is you care to work on. If there's a technological side to it, you work with the technology that's available, and you also work with the technology that you can afford.
Yeah.
And I mean, just by way of just sort of fairly recent history, back in 2013, we published the first genome of a turtle. It was a painted turtle. And if you looked at all of the different pieces of information and stuff that different labs donated to it, it cost about $1 million to sequence that turtle.
Wow. Yeah.
We can now, it's resequencing, which is a little bit of a cheat, but you can now resequence a painted turtle, its entire genome, for about $300. And that's since
10 years.
It's in 10 years. Right. So 10 years ago, you could sequence a turtle. 12 years ago, whenever we actually did the work before we published the paper. But could you afford, I mean, again, just to take an example, we're just finishing up a project with someone else who I think you've interviewed, Peter Scott, when he was here in the lab on Western pond turtles. And we collected really good genomic data on 1,600 pond turtle samples from all across the state, all across their entire range from Washington to Baja. Well, we can do that because it's gotten cheap, right? 10 years ago, that would have been 1,600 billion, right, or $1.6 billion
Yeah.
to
Right.
nobody's going to fund that, right? You can go to the moon for that. So it's a combination of the technology that's available at the time and what you can afford. So when we first started working on turtles, turtle phylogenies, we published, I think, our first paper back in '97, which I did with, a lot of that was also working with a couple of undergrads in the lab. And the sequencing technology that was available was really single gene sequencing. And there are all kinds of issues that are just problematic when you're trying to understand the relationships of organisms, who's most closely related to who and who's more distantly related. There are huge problems when you just use one character set, right? I mean, just imagine, morphologically, you look at, those of us, on this call right now, and if we use hair color, or if we used height, or if we used anything which has a genic basis, and therefore has some heritability, and therefore has some information about who's related to who, we'd get it wrong.
Yeah.
Right. And almost certainly, because single characters they have their own idiosyncratic histories.
Or it would give sort of a different interpretation.
Well, it would tell you about the history of amongst us on this call. It would tell you about kind of the history of our hair color genes, or the history of the genes related to height with respect to us, and those may or may not have much of anything to do with how we're related to one another in terms of our true genealogical history, going back from parent to offspring, parent to offspring, parent to offspring. And that's what we're trying to understand with phylogenetics. And so the idea basically has always been that if you use a lot of different characters, some of them are shaped by natural selection, and so they tell you a lot about where organisms live environmentally and less about their evolutionary history, about their phylogenetic history is related to. Some tell you about phylogenetic history. Some have one history. Some have another history. And if you kind of have enough of them and you average it all out, the one thing that they all are subject to is phylogeny. I mean, there is a true, set of relationships. If you have three species, two of them shared a common ancestor more recently with each other than either did the third. That's true.
Right.
And if you get enough genes or morphological characters or whatever, usually that signature comes through. It sort of all comes out in the wash, but you need a bunch of them. And so we have gone through different iterations as the technology has changed and as things have become more and less expensive of putting together different collections of types of characters that we can use that we think give us an accurate, reasonably accurate estimate of who's related to who. And then the other thing that has really strengthened the whole business is putting together, really good statistical models that allow us to say not just that, species 1 and 2 are more closely related to each other compared to species 3, but also allow us to put some certainty on that. So allow us to say, that's what the data says, but we're really not very sure.
Right.
As opposed to saying, we're really quite sure. And that innovation, those statistical innovations have been absolutely the kind of bedrock of being able to move forward in the business, because it helps you understand better what you know and what you don't know.
Right. And that's sort of I think you're sort of highlighting the idea of independent lineage sorting and how the different genes tell different stories.
Different genes tell different stories, yeah. And different genes I mean, right, different genes do tell different stories, and they tell that both because of things like natural selection, that natural selection has acted on them in different ways to constrain them or accelerate their rate of change. And they also do it through just these really interesting random, or as people say, stochastic processes, and like lineage sorting and lineage sorting is one of those. And I mean, I don't think this podcast is the time or place to go into those
The specifics.
just how that works, but it's a but you can get you can get lots of sort of disagreement between different genes and different parts of the genome in terms of how individuals are related to each other. And I mean, the one thing I'll say on that, a little techie thing, but
Yeah, I think that works.
Yeah, is just to say, so imagine you've got 3 species of turtles, yeah, or anything else. And, again, as we say, 2 of them shared a common ancestor more recently with each other, so than they did with the third. And that's the game in phylogeny, is trying to figure out which 2 are more closely related to each other compared to the third, or which 2 share a common ancestor more recently with each other compared to the third. And so if you think back in time, there was a single ancestral population, and it split into 2. Okay, and that leads to one species and another. Those now become 2 species. And then one of those, but not the other, split again and formed 2 new species. So that would give us the phylogeny, looking backwards in time now, of those 3 species. And if that time between the first speciation event and the second speciation event is long, so if the first split happened 10 million years ago and the second split happened 5 million years later, then if you think about it, that means there's 5 million years when those 2 species that derived from the second event, when their common ancestor diverged from the other one. And those changes that occurred are going to be shared between those 2 species. So if that internal branch, as it's called, is relatively long in time, it's a much easier phylogenetic problem. But just imagine if the first speciation event happened 10 million years ago, and the next one happened 9.9 million years ago. There's just a little itty bitty time in there. And there's just not much time for things to evolve on that little branch. And that evolution on that little branch, that's the information that's going to tell us that those other 2 are more closely related to each other than they are to the third. So a huge amount depends on exactly what happened, right? And, sometimes the world changes, right? We get a glacial or an interglacial, or, a meteorite crashes down and a lot of stuff goes extinct, or whatever happens. And oftentimes after those events, there's a lot of rapid bursts of evolution. And that's fascinating to understand, but it makes it really tough to figure out what happened in there, because everything was going really, really quickly.
Right.
Speciation was happening, boom, 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 boom. Those are really hard to separate out. Whereas, if the world is nice and stable for a period of time, well, then, speciation happens for one reason or another, but those internal branches oftentimes tend to be longer, and those tend to be much easier bits of history to sort out.
That sort of highlights something kind of interesting. I think that there was the hopeful monster hypothesis, and that's one that was based on developmental studies more. But based on what you're saying, that would be tough to pick up in a phylogenetic framework, because that sort of hinges on the idea that there were rapid bursts of change.
Well, I mean, hopeful monsters, you may or may not know this, but the other group of organisms of amphibians that I study a lot are salamanders. And And sort of the quintessential hopeful monster in many ways is this thing called an axolotl. It's a species, Embysma mexicanum, that occurs in Mexico. I did my PhD work on them. I've continued to this day studying their relatives. California tiger salamanders, a federally endangered species that I work on a lot, are very closely related to axolotls. And so what's an axolotl, right? So an axolotl is a larval salamander. A lot of salamanders, not all, but a lot of salamanders have a larval phase that lives in the water. They go through a metamorphosis, they turn into terrestrial phase, and then that terrestrial phase is what we think of as the adult salamander. Same with frogs. The tadpole or the polywog turns into an adult frog through metamorphosis. Well, what an axolotl is a larval salamander that never metamorphoses. It turns into a sexually mature, large larva. It's got gills, it's got a tail fin, its skin never changes, it doesn't get a color pattern. If its pond or lake dries up, it dies. That is, we've done work on this, other people have. There's some genes associated with the metamorphosis hormonal cascade for thyroid hormone and its receptors that gets interrupted and leads to that new morphology. So that, is a hopeful monster in the sense that you get this thing that in one swoop with one mutation, you get a lineage that doesn't look anything like its ancestor. They're just completely different, right? One is aquatic, one is terrestrial, one has gills, one doesn't. One takes 4 years to reach sexual maturity, one takes 6 months. Everything about them is different. And the hopeful monster, and maybe this is what you were saying, the hopeful monster transition, that is true, happens very quickly. In that case, because it's a single developmental gene. So I guess that and working out those animals has been extraordinarily difficult from a phylogenetic point of view. I think finally got that worked out, but it's been very hard, very slow process. So you're right. The hopeful monsters are derived from their less monstrous relatives very quickly, and so that is a difficult phylogenetic problem to sort out. Not all instances of developmental change happen quickly like that. Sometimes developmental evolution is slow and gradual, just like other forms of evolution. But you're right. Now, are there any hopeful monsters in turtles? I don't know.
Yeah, I
don't know, but fair enough. Anyhow.
Yeah, well, I think that's it. We definitely want to talk about sort of the turtle phylogeny and how the nodes in the turtle phylogeny have changed. Beforehand, though, there's some, and we sort of touched on this. There was a paper that you wrote on this, troubleshooting phylogenies, and that really points out how diverse the approach is. And this was a while back, and stuff has certainly changed. But when you're dealing with more historical, now we've got genomes, and there's sort of a different type of analysis for that. But with more historical phylogenies and tree construction, again, the beauty of that is how complex it is, and where all the different variables and data come together and produce a tree. At the same rate, with more sort of model complexity, you get more things that could go wrong. And there's some things that are pretty consistently can go wrong in analyses. And you've touched on the idea of the sort of marriage of statistics with the phylogenetics. Maybe you could explain sort of how that kind of the bootstrapping analysis, how that works. What is that telling us about the accuracy of a tree?
Sure. Well, if you want to understand bootstrapping, which is one form of assessing the sort of reliability of a tree or a node on a tree. So bootstrapping is a general it's a general strategy for putting probability values on statistics. And the way it works is like this. So first of all, what's the logic behind it? So let's say you're using genetic data, or let's say you're using morphological data, either one. There's a very large array of possible genes that each have sort of different histories, or possible morphological characters that are potentially available for an organism, right? You picked some of them, maybe because they were easy to get your hands on, maybe because that's what the technology allowed you to do at that time. But up until recently, at least with genomics, you seldom had the whole thing. You seldom had all the information, and even if you did, you didn't have every individual. So you have a collection of individuals that you collected a collection of data on. And what you'd like is to be able to believe that collection of data and individuals that you have are really representative of the possible data and the stories that those individual pieces of data, those individual characters can tell, and the possible individuals. And you might have some collection of data that is biased in certain ways. You might not be sure about that. And so the way that bootstrapping works is, so consider the following. So let's say you have 100 nucleotides worth of data, 100 positions, and they're variable, and we've got that across a bunch of individuals. Well, if that 100 that you have is a reasonable, is a random set from the genome, from those 2.6 billion that you could have found, okay, then that's good. So you take that random set of 100 characters and you analyze it once and you get a tree. Okay? But what you'd like to know, the experiment you'd like to do is to say, let me go back into the genomes and get another 100. And let me build the tree again. And let me see if species A and B, or individual A and B, are each other's closest relatives in both of those. And what I'd like to do is do that again, and do that again, and do that again. Do that a lot of times. Do that 10,000 times. And if every time species A and B are each other's closest relatives and species C falls outside of that, then I'm going to feel pretty darn confident that the right phylogeny is that A and B are each other's closest relatives. Okay, if I only do it once, or maybe there's just something weird and idiosyncratic about that particular set of 100 base pairs. Well, normally you can't do that. You can't go back into the genome, and especially in earlier history, you couldn't. You had your 100 base pairs, and that's what you were stuck with. Okay, so what could you do instead? Well, rather than going back into the genome and pulling out 100 new independent characters at random and redoing the exercise, you can say, well, if my first 100 is a random set from the genome, then what I can do is take those 100 characters, go into that set of 100, and pull one out at random. Okay. Now, take the same 100, go back in and do it again, and do it again, and do that 100 times. Okay. And what you're doing is, and why isn't that second set of 100 identical to the first set of 100? Well, it's because you're sampling that first set of 100, as they say, with replacement. Okay. So what it means is that you might get nucleotide number 27 on the first draw, just doing luck, and I'll stick that in my data set. And then you might go back in and get it again. right? You might happen to hit it just by flipping a coin. You might happen to hit that 2 or 3 or 4 times, and there might be several other characters that you miss just due to luck. Okay, and that will happen. There's big statistics behind that. And it turns out that if your first set of data was a good random draw from the true distribution you care about, in this case, the genome, then each one of those 100 samples where you sampled with your placement will be an equally good representative draw as if you had resequenced it, you had gone back into the original genome. And so it's a way you can leverage a small data set. And then what you can do is for each one of those bootstrap samples, you can say, let me rebuild the genome, okay, with, some characters sampled more than once, some not sampled at all. They're all different. And it gives me the opportunity to now do that 10,000 times and say, how often are species 1 and 2 related to each other? And, what you can sort of see, the logic of it is if you have very strong support for A and B being closely related with respect to C, then what does that mean? That means a lot of those 100 characters are going to be shared by A and B and different in C. And so when you go back in and resample, you're always going to hit those over and over and over again. And if instead, there's 10 characters that say A and B are each other's closest relatives and 9 that say A and C are, well, then in those bootstraps, you're going to get lots of both of those. And so those trees are going to be very discordant, and you're not going to get very strong support for any one phylogeny.
Right.
So anyhow, that's how it works.
I think that's a good thing to cover before going into it, just because it's so critical, as you said, to understanding the accuracy and sort of
it is
interpreting results.
Yep.
So with that, there's issues in other respects in terms of tree topology and that sort of thing. But I'm kind of curious because, as we've mentioned earlier, you've studied turtle phylogenetics for at least 30 years, I would assume. Yeah. So you've seen how these things have changed, but you've also seen the actual phylogeny, the most likely scenario. You've seen that change over time. And in terms of the nodes of the turtle tree, I'm sort of curious, what are the biggest changes you've seen that maybe have surprised you, and why? What's the methodologies that underlie the new understanding?
Yeah. So remember too, with phylogeny, or genealogy, you can study phylogeny at sort of any level in the hierarchy of the tree of life that you wish. So you can study within turtles. You can take, say, the 13 generally recognized families of turtles, and you can ask how are they related to each other, but not care about the species within the families. I just want to know, who are sea turtles related to, right? There's 2 families of sea turtles. Are they each other's closest relatives? How are they related to other turtles? You can also ask the question, at the species level. So you can say the Western pond turtle, which we've split into 2 species, who is it most closely related to? Within that subfamily of the family that's in, there's a few candidate species. How are they related? You can do it at the population level. You can say, are Western pond turtles from Orange County and LA County more closely related to each other than either of the most San Diego County turtles? You can do it at any level or in time frame you wish. When I started working on turtles with Peter Nayland, who I think you've also interviewed. Peter and I were old friends from work we did when I was an undergrad and he was a graduate student years and years and years ago. But the thing that we tackled in that first bit was, how are the families of turtles related to each other? And we did that using a couple of genes and some morphological characters, and then we threw in some fossils to try to understand the time course of that, because Peter was basically a paleontologist. And we came up with some answers and also some confusions. The most at that level of the families of turtles,
It's sort of global phylogeny.
global phylogeny, so all of the species and all the families of turtles across the world. The probably two most perplexing ones, the ones that were just the toughest to figure out, were snapping turtles, which is really interesting. Snapping turtles. There's alligator snapping turtles and common snapping turtles, and they are super widespread. And common snappers are across most of eastern North America. They can be very abundant. They're very heavily exploited in the food markets and trade, often to a very unsustainable level. And if you know what a snapping turtle looks like, they're enormous. They're just bristling with all kinds of weird characters. They have these super long spiny tails.
Yeah.
They have a little tiny plastron, a little tiny bottom shell that's very, very reduced and hardly even connects to the carapace. They're very aggressive. They have huge hypertrophied heads. They're just weird turtles, right? And so no one has ever doubted that snapping turtles are their own group. And alligator snappers and common snappers are very similar. There's a couple of other species in the common snapper genus, Calidra, that occur in Central and South, or in Mexico and Central America and South America. No one's ever doubted that that's a group because they're so weird. But because they're so weird, because they have so many unique features, it means they don't share those features with a lot of other turtles. And so it makes it very hard to figure out who they're related to. So for that, you need shared similarities. So, and that's a generalization, right? I mean, whales, right? I mean, no one doubts that whales are a group, right? But they're so weird. Figuring out who whales are related to has been a really hard problem, right? And that's just sort of generally true in phylogenetics, that easier it is to recognize a group because they're so different. The harder it is to figure out who they're related to because they're so different. They're different from everybody else, so that makes it hard. So anyhow, it turns out whales are closely related to hippos. Right? That's one of the coolest phylogenetic results, I think, to emerge in the last 20 years. Well, who the hell are snapping turtles related to, right? It's really been a tough problem. And I feel very, very confident that with using, very large genomic data sets, we finally have figured that out. And the answer is that snapping turtles are related to mud and musk turtles in the family Testudines. And, when you look at them, some musk turtles in particular have, very reduced, plastra. The weird thing about it is that most mud and musk turtles are really little. Most of them are, I don't know, 5, 6, 7 inches long. They're small turtles. Not all, but most of them are. And snappers are huge, right? So based on that character, you'd be kind of very surprised to think of them as each other's closest relatives. But the data are pretty unambiguous. And I don't think pretty. I think the data are now at this point completely unambiguous. And so, that's one example. Of something that has just been a persistent problem for years. And one really interesting thing is that there was a guy who was on the faculty at Harvard University, who's the herpetologist in Harvard's Museum of Comparative Zoology, the MCZ. His name's Ernest Williams. And if you know herpetology, you know Ernest Williams as the person who sort of invented anolis lizards, the American chameleons, they're sometimes called. Fascinating radiation of lizards that have speciated all through the Caribbean islands and all through mainland Central and South America. And Williams really generated the anoles as a model system in studying evolutionary ecology. Well, Williams did his PhD work on the evolution of the cervical vertebrae, the neck vertebrae of turtles. That's what he did his PhD on.
That's where I think most of us and our listeners are familiar.
Oh, we're familiar with
turtles,
Williams.
right?
Okay, well, most people
Yeah.
don't know that about Ernest Williams. And if you read the monograph, which was his PhD, Ernest Williams says, snappers are a big problem. And he said, and based on their neck vertebrae, they're related to mud and musk turtles. So Williams got it right.
Yeah, right. And that's not always the case.
That's not always the case, but Williams did.
It's interesting that line of morphological data was sort of corroborated what you found with multiple different lines of evidence. But the Platysternidae, that's another one that was sort of an issue. Maybe you can speak to that, but you would think that just based on gross morphology, they're similar to snappers, but that's
and Platysternids are big-headed turtles. That's their common name. Another, either one or a couple of species, depending on your taxonomic preferences. With Asian turtles, small, long tail, big head, but a very weird big head with all this secondary ossification in it. They climb. The reports, I've never seen a Phylogenetics in the wild, but the reports are that they climb low trees and bushes, and they live along streams systems for the most part, all through Southeast Asia and parts of South China. And again, as you say, at a very superficial, at a very morphological level, they've got that big head, they've got that long tail, they've got some of the features that define Phylogenetics and snapping turtles, and morphologically they were put together as sister taxa and as sister families. And it turns out that Platysternon, is the sister group to amidid turtles, to freshwater pond turtles, and chelydrids, snapping turtles, are the sister group, the closest relative to mud and musk turtles, and those two are in very different parts of the turtle phylogeny. So what the molecular data has shown is that the kind of gross similarities between big-headed turtles and snapping turtles is convergent. It's not indicating that they're very closely related. It's indicating instead some common features of their biology, which, to tell you the honest truth, I don't think we understand.
Yeah.
I don't think we understand why they are so, morphologically similar.
Right. It is,
but it's independently derived. It does not reflect the fact that they have a very recent common ancestor.
Yeah, that's something to speculate on, and for people listening, something that you could definitely spend a lot of time trying to figure that out. There's one more sort of node that's been challenging, and then maybe we can talk a bit more about genomes of turtles. Sure. But before we sort of move on to that, the trinecoids, the soft shells and allies, that's one that even today is sort of ambiguous.
Well, so again, presumably your people know this. The major split of turtles, right, is between the side neck turtles and the hidden neck turtles, the cryptodires and the pleurodires. And more or less, hidden neck turtles, which pull their head in with a vertical S curve, and are cryptodires are more or less northern hemisphere turtles, and pleurodires, which pull their heads into the side, like a monomoto turtle or an Australian snake-neck turtle, are more or less southern hemisphere turtles. And that kind of goes back to the makeup of Gondwana and into the different major continental groups. So cryptodires and pleurodires, everyone has always agreed, are are very separate ancient lineages that diverged a long time ago, and that's the molecular data has all held that up. Soft-shell turtles are cryptodires. So they based on how they pull their neck in, with the vertical hinges. But they're very weird turtles, right? I mean, have tight scutes on their shells. They're a very strange, bizarre group of turtles. And what goes along with them pretty clearly is pig-nosed turtles or fly river turtles in the family Cretz, which are restricted to New Guinea and Northern Australia. And that goes with the soft shells. And so the kind of question has always it's always been pretty clear that soft shells really go in with the hidden neck turtles, with the cryptodires. But exactly where and how, and again, it's just because they're so weird. They're just so divergent. A lot of the characters that we use to link other turtles together, they've just lost them or modified them so badly you can't even see them anymore. And what has really kind of emerged is that there really are three major lineages of turtles. There's the side neck turtles, there's the non-soft shell cryptodires, and there's the soft shells. And what the molecular data has informed us of, I think very unambiguously, is that yes, soft shells are cryptodires, but the first split in the evolution of the cryptodira was between soft shells and everybody else. Yeah. And that everybody else includes marine turtles, includes leatherback turtles, it includes, snapping turtles, it includes big-headed turtles, it includes tortoises. And so the split between soft shells and all of the others, sometimes called the Duracryptodira, the hard-shelled cryptodires, was a very early split that happened not too long after the split between pleurodires and cryptodires. So it's a relatively short internal node.
So they're a very ancient one.
They're a very ancient clade. They're a very ancient clade. And the first split in those is between the pig-nosed turtle, which is a single species that is just hanging on as a single species, and the other, whatever we're up to now, 27 or 28 species of soft-shell turtles, which are distributed across most of the northern hemisphere. And interestingly enough, if you look at a combination of evolutionary uniqueness and sort of conservation status based on just population status, the pig-nosed turtle is probably the most endangered long branch of turtle evolution in the world. So they're very heavily exploited, and the way to put it is that if we lose pig-nosed turtles, we lose as one species. We lose more of the entire history of all turtles on Earth than any other species of turtle.
That's an interesting 150 million years.
Yeah, it's a long piece of evolutionary history, and there's no other survivors from that lineage. So we're down to just the pig-nosed turtle. So anyhow, it's an important one. But the placement of soft shells has always been tricky, and it's very solid now. Yeah, we understand it very clearly, but there are old hypotheses that said that soft shells were closer related to mud and musk turtles, and turned out just not true. And it's interesting to go back to some of the morphological characters that people used to link those and say, well, what's up with those characters? Why are there some sort of subtle similarities? But that's a different thing in terms of just understanding how those speciation events led to the phylogeny that we have now. It's a very ancient split within the cryptodires.
Right. It's
and there's still.
Go ahead.
Yeah.
Sorry.
There's still disagreements within the soft shells. So, like for example, as far as I know, it's still not certain if Apollonia mutica is sister to Ferox or Spinifera. I wonder if you have reservations for
I
either of
think
those theories.
I think the Ferox, the Ferox spinifera connection to the exclusion of mutica is now really quite well established. That's
Right.
it has been and there's, I don't know how well you know those turtles, but things like the nasal septum, which is the character that I always use to tell a spinifera from mutica, and Ferox shares the nasal septum with spinifera. They're I'm sure there's other characters. That's the one I happen to use, but the molecular data is very strong on that. So I think that's really become pretty well resolved at this point.
Okay.
Yeah, soft shells have always been a tricky group to understand both species boundaries and phylogenetic relationships. A guy who got his PhD in my lab years ago named Tag Engstrom did his PhD work on the phylogeny of soft shells, and I think made pretty good progress. As you probably know, Peter Malin, did his PhD on the phylogeny of soft shells, and he was the one who first broke them up from being a single genus, from all being in the genus Trionyx to whatever we have now, 8 genera or something like that. So Peter, I think, made some big strides based purely on morphology. I think Tag made some additional big strides bringing molecular data into the story. But there are still some tricky ones, and I think the big emerging question now with soft shells is species limitation. So things like Pelodiscus, if you know them, the Chinese soft shell, always considered to be a single species. Now Sinensis has been broken up into, I can't remember how many, but 8 or 10 species, and testing and validating those species, understanding their geographic ranges, understanding how they're related to each other within the genus Pelodiscus. I think those are some of the big challenges right now. Same with some of the sub-Saharan African soft shells.
And is the Rhaphodus and Apollon relationship still upholded?
Yeah,
Previous.
I think
It seems really interesting to me.
it is really interesting. So, and Rhaphodus itself, which has a very disjunct distribution. One species that's nearly extinct in South China and Southeast Asia, and the other in the Mediterranean. Is the fact that those two are sister groups, I was sure they wouldn't be. I just didn't make any sense to me. Why should they be? But it really looks like they are. And then the relationship of Rhaphodus to Apollonia. It's actually I'd have to look at some of my own work to tell you. But my memory is that's held up pretty well.
Okay.
Which again was originally proposed based on morphological characters, and I think has held up. Yeah, but and again, hydrographically, what's going on with that? Those then become the very interesting downstream things that we can study with those phylogenetic results, right? To understand how that relates to continental drift, or one of the things about soft shell turtles, again, a little odd in my mind, given that they don't have a hard shell. You would think they might not be very good at dealing with salt water, but like Trionyx triangulus, the soft shell of the Nile soft shell, they got those apparently 100 meters down in the Eastern Mediterranean, eating clams, right out in the Mediterranean Sea, and soft shells are actually pretty good at making it in salty water. Some of the giant Asian soft shells are really good at being in briny salty water. So maybe that's a part of the funny disjunct distribution of some of the soft shells as well.
Right. Well, it's good to know that all this these additional markers that are being used have helped to resolve the relationships in soft shells, because that's not always the case. More data is always better than less data, but sometimes it doesn't help to generate more resolution in the phylogeny. And one
Yeah,
of the solutions that you write is bacterial artificial chromosomes. I'm interested in that because I've used like triple digest libraries to sequence, my toads, but I've never heard of your BAC approach. If you could talk about that, that would be interesting.
yeah. Well, the back libraries are at least in my understanding, um, they're they're I think they're falling out of favor. So this is almost a little bit more historical
Mm.
than it is current. So the the sort of logic, okay, so when you think about if you want to assemble a genome, okay, so you want to get the whole genome, you want to get all the chromosomes, and you want to assemble them all in a linear array as they exist in the cells of the body of the organism. Okay. So what do? Usually sequences are short. So you take it, you can, it's easy to, as you know, extract the DNA from an organism. You shear it, you chop it up into pieces, okay, and that's random. So all the pieces are there. And the analogy that we all use, because it's a good one, is jigsaw puzzles. So all the pieces are there, and they've got to sort of put them all back together. You got to sequence them and then put them back together again to get those chromosome length arrays. And the sequencing is usually short, right? So most sequencing is in 150 to 500 base pair pieces that you've got to assemble into chromosomes that might be 100 million base pairs, or 200 million or 300 million base pairs long. And so the way you do that is you just take all of those fragments that you've sequenced, right, and you look for overlap. You look for a 20 base pair unique sequence at one end of one fragment that is also found in a different fragment, and now those two are the same, and so you can stick those together, and now you got a 600 base pair piece, and then you find another one and stick that on, and now you've got a 900 base pair piece. And you sort of build those out, and those are called contigs because they're contiguous pieces of DNA, and you bioinformatically build those up, and those get you, depending on the sequencing that you did, to a certain point. And now you want to glue those together, right, into what are called scaffolds. And those are saying, okay, I've got a contig and another contig, and what I'd like to know, even if I don't have the stuff in between, I'd like to know where they sit relative to each other and how far apart they are. And so the way you do that is you somehow generate from a long piece of DNA, a piece of DNA that's 5 or 10 or 20,000 base pairs long. You generate some sequence from those, and then you say, okay, well, I've got this 5,000 base pair piece. So what a back library is that you take the DNA from your organism and you break it up into big pieces, like 5 to 10 kb, 5 to 10,000 base pair pieces, and you insert those into bacteria, and you can use then that construct, the bacteria, to grow them up and to replicate them. Okay. And what you can do, just the nature of that construct, is you can sequence in a few hundred base pairs from the end of each one of those long pieces. So you sequence in 500 base pairs or 400 base pairs in to the ends. Okay. Mm-hmm. Now what do you have? Now you've got a sequence, a 400 base pair sequence, and another 400 base pair sequence, and they're connected. They're in the same piece of DNA. So you know how long that piece of DNA is. So you know that those two bits are on the same scaffold. Now you can take each one of those 300 or 400 base pair bits where you in sequence that back, and you can look across all of your contigs and say, do I see the left-hand one anywhere? And if you do, I say, okay, the contig that I've got, it goes there. And then look on the other end and do the same thing. And so that allows you to build out these bigger scaffolds, right? These long, you don't know what's in the middle, but at least you know that contig 1 and contig 2 are on the same stretch of DNA, and that there's about 4,000 base pairs between them of something unknown. If you want, you can sequence your way in, but that's very time consuming. But that's what BACs were used for, is to create scaffolds that allowed you to take your contigs and glue them together into longer pieces, okay, that start to feel more like chromosomes. And that's just part of doing an assembly of a genome. Well, now we can use different technologies for that. So now you can do single molecule sequencing that's 10, 15, 20,000 base pairs long. So you kind of don't need the back anymore, because you can just take the raw native DNA without putting it into a bacteria and growing it and plating it out and stuff, and doing all that stuff that we had to do 20 years ago. Now, the sequencing technology is such that if you have a really, really high quality tissue sample or blood sample that went, from the turtle directly into liquid nitrogen, and and it's really intact and is really high molecular weight DNA with really long fragments in it, you can just sequence those. And you can do that, PacBio hi-fi sequencing, for example. Those go up to about 15,000 base pairs. And the brand new Prometheon sequencing, if you're familiar with it, you can go up to, I think you can go up to about 50 kb, about 50,000 base pairs on a single molecule. And in fact, we're just a couple of colleagues, guy who you might be interested in talking to, named Caleb Krumer, who's a grad student at Michigan State University. We're collaborating on using that new Prometheon sequencing, that super ultra long sequencing on a spotted turtle, to create what we hope will be the first true chromosome level assembly of a turtle. And we're doing spotted turtles, Plymouth guttata, simply because they're so heavily exploited in the U.S. They're my understanding is they're the #1 confiscated turtle at LAX and at JFK, for going out illegally for the turtle trade. And so it's part of a project to sequence spotted turtles from all across their range in the U.S. and anyhow use this reference as part of that project. We can talk about it if you want. But anyhow, so going back to all of this, those back libraries were a technology that, as it turns out, was kind of an intermediate technology that allowed us to scaffold together those reasonable long reads that we could put together. We used it in the painted turtle genome, for example. Right. And that's now been sort of supplanted by these longer single molecule sequencing techniques that allow you to do it in a much simpler and much more effective way.
Right. I always feel glad that I live at a time where these genomic methods are readily available, because Sanger sequencing is such a pain. Even if you have like just 5 loci, you have to calculate the annealing temperature, and half the times it doesn't even work.
No, trust me. I mean, go back and look at that 1997 paper that we did, across turtles, because one of the things about Sanger sequencing and annealing temperatures and trying to get the primers to sit down is that that varies depending on the sequence, right? And turtles at 220 million years of divergence, even at a slow rate, there's a lot of evolution. And so, you had to make taxon-specific primers and try and get them to sit down in different places. And it was a big lift to try and get that to work, even just, for cytochrome B and 12S, just a couple of easy genes, and across that kind of divergence. And now, all that is ancient history. And I agree with you. We're now we're routinely, resequencing entire genomes of hundreds of individuals, and we can, thousands in some cases, and we can do that now. And 5 years ago we couldn't, and right now we can do it. You obviously know a lot about genomics. We can afford to do it at roughly 10x coverage, which means you're kind of right in the zone when you can accurately call heterozygotes, so we can understand heterozygosities, and we can, which is a very important concept for studying inbreeding depression, which is a major problem in a lot of turtles, or I think it is. But trust me, by the time you've got your PhD, you're going to look back at us doing things at 8x and 10x coverage, and you're going to say, pathetic, because now we can do it at 100x coverage, and we can call every SNP and every variant and every heterozygote with absolute certainty, and that will be absolutely doable in 5 years.
Right. It's not just the how easy it is to sequence a genome. It's also just simply the more accessibility to the nuclear genes, because we've seen so many species descriptions that have been made out of mitochondrial DNA, and those those aren't really good at all because of the the maternal inheritance of mitochondrial DNA.
Sometimes they are.
Sometimes they are. Yeah.
How do you know?
Mhm.
Right? Sometimes they are, sometimes they aren't. How do you know? I have always thought, you guys should do this. We're probably at a point now where we could do this. Is that for taxa where we have mitochondrial DNA, good mitochondrial DNA sampling, and taxonomic decisions were made based on that, and we now have good nuclear DNA sampling, and ask what fraction of the time was the mtDNA wrong, and it's not wrong, it's just telling us about its own history as opposed to the history of turtles, right?
It gets misinterpreted sometimes, because like in lizards, like in Scaloporus, there's lots of mitochondrial ingression, and then we don't recover the actual population lineage.
Yeah, and in
just something you have to use cautiously.
In turtles there is too. But wouldn't it be interesting to now ask what fraction of the time was it and or what fraction of the time did we legitimately and reasonably misinterpret it?
Right.
In other words, again, it's not that it was wrong, but we interpreted it as telling us the history of the organisms overall, whereas in fact it only told us about the idiosyncratic history of that. You have the mitochondria, and I don't know what the answer is. I have no idea if that sort of error rate, if you will, is 5% or 80%, and I don't think we know. And I think the data are starting to be there. They're sitting on GenBank.
Yeah. Do you have any specific examples of mitonuclear discordance that comes to mind in turtles?
Well, one example on the books right now, and we're really trying to it may not be as big an example as we thought it was, is with Western pond turtles. Mm-hmm. So Western pond turtles, which are currently recognized as two species, the genus has been up for grabs for a long time, but we've really settled in on Actinemes as the appropriate genus. And there's Actinemes marmorata and Actinemes pallida, pallida being the southern species, marmorata being the northern species. And the question area has always been the central coast of California, roughly from Santa Barbara to San Francisco, and sort of which one is in there. And based on currently published data, which is all pretty much come out of our lab, mitochondrially, the central coast is the northern species, marmorata, and nuclearly, the central coast is the southern species, pallida, which goes up to
Right.
at least Monterey and maybe up to San Francisco. And so there's clearly been some, fairly serious mitochondrial nuclear discordance there in that large chunk of the area of the range. And I mean, new data that Peter Scott and I and a couple other people in my lab have pushed out, looking in much greater detail at the central coast, says, well, maybe there actually is a little more hybridization in the central coast than we had thought, and so then that discordance isn't perhaps quite as discordant as we had thought. So it's still an evolving story, but at least there's clearly been a fair bit of discordance in that region. Maybe not quite as much as we had thought, but with the big RADSEQ data set, I think we're finally coming to a resolution on that. But that's one example. Can I think of any? Oh, well, yeah, a great example of mitonuclear discordance is at a higher level. So the subfamily Amidinae, so the Amidids, right, the sort of basically the New World, North American plus a little bit of Europe, pond turtles are split into two subfamilies. They're the Diarchelines, that's painted turtles and layered sliders and that kind of stuff. Genus Pseudomys and Dracomes, and anyhow. And then the Amidines, and the Amidines are the western pond turtle, spotted turtle, wood turtle, bog turtle, and the European pond turtle, Anis orbicularis and Trinacris. And the European one has always been sort of an enigma, right? It's what's it doing over there? It's the only Amidid and the only Amidine in Europe. And so if you think about it, that subfamily of Amidinae has one species that's restricted to the west coast of North America. It's got a handful of species, also Blanding's turtle, which I forgot to mention, that are in the Great Lakes to the east coast. So there's one species in the west coast, one species essentially in the east, and one species in Europe, or several species in the east, excuse me, but three different groups. And the question is sort of how are those related to each other? And the key players, this is clear, are the western pond turtles, Blanding's turtle, and the European pond turtle. So those, more or less three lineages at least, 5 species or something, form a clade. And the question is, which two of them are more closely related to each other than the third? So is Europe more closely related to the eastern US than it is to the western US? Or are the two US ones more closely related to each other from the two coasts, with Europe splitting off first? So I don't know. I mean, do you know this story?
No, I'm
do you
not
guys know
familiar
this
with
story?
I've reviewed some of this, but it seems like the morphology, you would think 100% that it's Actinemes and Emis, but that it doesn't seem like that was the case.
Yeah. So
And
the the mitochondrial DNA said that let me make sure I get this straight. Actinemes and Emidoidea, so the two US ones, are sister taxa, with Emmys orbicularis, sister to that. Okay. The nuclear data says Emidoidea and Emmys. So the Eastern US and Europe are sister with Actinemys sister to that. And that seems to be the correct answer, but there's a big mitochondrial discordance because the mitochondria say the opposite. And
Right.
one of the things that, again, my sense, I've never seen a good review of this, but my sense is that frequently mitochondrial DNA, and I think this is just because it's this super gene, it's a single non-recombining unit loaded with important genes that are involved in metabolism and energy production and stuff. It's just loaded with these really important genes from a selection point of view. That mitochondrial DNA often seems to link together species that live in the same area. And again, if it is important and responsible for sort of survival in certain environmental conditions, then, and if there's any hybridization at all, then that mitochondria can sneak through that hybrid zone and be shared across those species that are in the same environmental conditions. And so I think mitochondrial DNA often seems to indicate monophyly of things that live near each other.
Mhm.
And it oftentimes and I think that's where some of the mitochondrial nuclear discordance that is out there is derived from. But I've never seen a meta-analysis of this. I mean, no one's done this to my knowledge. I think it would be a really interesting one. But I do think it's interesting that the mitochondrial DNA said the two species on the same, or the two lineages on the same continent were closely related, and the nuclear data, which goes more with the morphology, says that Emidoidea and Emmys are more closely related, and they used to be in the same genus. Yeah,
it's a pretty interesting idea. I like that idea of going back. It's evolutionary biology is a very forward-looking field, but taking a step back and looking at turtle phylogenies at all different levels, and more so at the familial level, and seeing were certain data sets correct most of the time, what percentage of the time. That would be really interesting.
Yeah, it'd be really interesting. And because again, like I think you were alluding to, we have in terms of just biodiversity generally, we have a tremendous amount of mitochondrial DNA data available on GenBank. It's certainly for vertebrates, it's astonishing. And all of the cytochrome oxidase 1, the CO1 data that's available as the barcode gene that people are using, tremendous amount of data from a biodiversity perspective. And if we could just believe it in terms of what it tells us about historical, phylogenies, we would be way further along in the world of understanding the tree of life than if we have to worry about it and challenge it and stuff. And again, it's an empirical question to some extent, right? How often is it misleading, and how badly, and are there certain signatures of that misleading condition that if we could sort that out, I think would be a huge win for just biodiversity studies in general.
Right. Well, we're getting up to the 90-minute mark, but we have a few more questions.
Okay, sure.
If that's
sure.
fine.
Okay.
The one thing you brought this up a few times. This one it's one that to me is really fascinating. The painted turtle genome, and so the landscape of that genome particular with respect to therapeutics for humans, it probably the SLC 2A1 gene.
Yeah.
That was one where for diabetes potentially, and for anoxia, it's upregulated.
Yeah.
And there were some other immune function analyses you did, and some other interesting things in some potentially even the pathway analyses.
Yeah.
I believe it was in that paper. But just all of that with respect to immune function, the potential therapeutics for humans. What do you think that the efficacy of that is? Do you think it's
yeah? I mean, some of it you do because it's interesting and you publish it in a time and a place. It's interesting and provocative and fun at some level. To think about those things. It allows you, so that paint turtle genome, we have 57, 58 authors, something like that. It's just bringing in all these different labs and all these different perspectives on that genome. And that in and of itself was a really, I think, a really great accomplishment. It was really interesting. It brought together a lot of different people. So I've maintained for 15 years, so I never would have done otherwise. In terms of using, very distant related species for therapeutics on humans, like turtles, or using genes associated with longevity or genes associated with their kind of very robust immune function on humans, it's a dangerous dance. There's been a lot of evolution between, because what you're doing, if you think about it again from a phylogenetic perspective, is you're kind of saying that that if you think about it, going from a painted turtle down to the most recent common ancestor of amniotes and then back up again to a human, I don't know exactly, but you're at 600, 700 million years of evolution, right, along those branches, something like that. A lot can change. Yeah. And a lot of things that work in one lineage don't work in another lineage anymore. And so the key to it, and I think the reason why the National Institutes of Health, for example, have been interested in fleshing out the vertebrate tree of life with whole genomes, is that the more that the mutations that we saw and the signatures of selection that we saw in the painted turtle, at least if for all we know, or knew then at least, those could have evolved in Chrysomenes picta, right? And be just totally unique to Chrysomenes picta and have nothing to do with the general longevity of turtles, right? Because we didn't have any other turtle genomes. Well, fill in some more turtle genomes and ask, do those characterize all of turtles? Now at least you've got a handle on saying that those evolved features are associated with long lifespans or associated with certain immune functions across a clade. Then ask, well, what about in crocodilians, and what about in squamates, and what about in mammals, and what about in eutherian mammals and non-eutherian mammals, and the more you can fill that in and see what the history of those changes are and how those orthologs, those homologous genes, are in those different groups, I think the stronger ground you're on for saying you can use them as therapeutics or use them to apply to a very disrelated, group. Because I think some you can and some you can't. Ultimately, comparative data is only going to go so far. You're going to have to do experiments and experiment with living model organisms where you can test those things. But I think at least filling in the phylogenetic tree with kind of key nodes is a really important part of that.
Right. It's maybe extrapolating from turtles to humans is kind of a jump, but if you can fill in the gaps,
If you
in
can fill in the
tree,
gaps and you can
Yeah.
sort of see what the changes are, and maybe that's a very, maybe there's some very conserved genes in there that haven't changed all that much, and those become much greater candidates for for therapeutics that you might want to use in humans or other mammalian systems, than ones that are evolving all over the place and evolving like crazy. Those might be less attractive candidates. So I think filling that in really helps a lot.
Right.
Yeah.
Yeah. That's
Yeah.
good to know. That's something that's real interesting for me, a lot of people
Yeah.
in terms of potential career pathways, that sort of thing.
Absolutely.
And
Yep.
and justification. Another thing too. So we talked a lot. I think holistically, we've talked about a bunch of different aspects. I have phylogenetics, population genetics, genomics, but there's kind of something that's outside. Not maybe 100% outside of this, but it's sort of a separate field. It's a newer field, epigenetics, and I'm sort of curious with turtles, what's known with respect to that.
Yeah.
Yeah.
And this is a place where I'm pretty much of a neophyte in terms of a lot of epigenetics work. I mean, I'll tell you one of the and the only application of epigenetics, and this is DNA methylation, which you might be familiar with, that we've just started exploring in my lab, is that so methylation, which is just attaching methyl groups onto parts of the DNA, and it controls the expression of genes. It controls whether certain parts of your genome are going to be turned on or turned off. And methylation, there's this concept of a DNA methylation clock. So within the lifespan of an organism, it's sort of a constant fraction of its genome tends to get methylated over time. So two cool things about it that I think would be very, very relevant to things I'm interested in, and with turtles as well as other organisms, is that if you understand that DNA methylation clock, and if it's and it appears, based on my understanding of it, there's a real expert in this, guy named Matteo Pellegrini, who's in our molecular and cell biology department here at UCLA. And Matteo's a real leading expert on this. That clock is apparently pretty constant for different species. And what that means is that you go out and you study some population of turtles, and you'd like to know how old they are. Okay, is this sort of and especially with turtles, where we don't tend to see a lot of baby turtles, and oftentimes they're either in different microhabitats and we miss them, or they're all getting nailed by raccoons and skunks and rats and other nest predators. And so we have this notion of these kind of aging populations where everybody, there's still a lot of turtles around, because they live for so long, but they're all old. And if there's not replacement going on, if it's a sort of unhealthy demography, then in another 30 years, that population is going to be gone because it's not recruiting. Well, there's all the difference in the world between those turtles being in a range of 10 to 50 years old, and there's just sort of, we're just not seeing new recruits. Maybe they come into the population slowly, but at least for adults, it's an even age distribution, as opposed to that, they're all 40 years old, because there hasn't been any recruitment in 40 years, because that means they're kind of heading towards the edge of the cliff. And the DNA methylation clock, at least in principle, allows us to figure that out simply by getting a sample, a drop of blood, and being able to study that. The other thing that is really cool with it is that the rate of DNA methylation within organisms, in other words, the rate of that clock increases with stress. And so something I've always wondered about is, we have, for example, urban populations of turtles and lizards. Are they under a lot of stress? Are they, when you're stressed, you tend to be immunocompromised, your immune system doesn't work as well, you're more susceptible to diseases, all that kind of thing. If they're living in dirty water, if they're living in, with very small population sizes where there's a lot of inbreeding, is their DNA methylation clock even more sped up because they're living under stressful conditions? And that might mean they're in even more trouble than we understand. So anyhow, those are two places where at least DNA methylation, which is a form of epigenetics, have been important. I'm sure there's lots of others. I'm just not very educated on them.
It's like a way to sort of quantify how well the population is doing.
Yeah. Yep. And we've got a couple of populations, not on turtles, but on toads and fence lizards, to look at that right now. A couple of new projects in line.
Something I know Jack, who couldn't make it today, was pretty curious about is the sort of the skull restructuring and megacephaly that occurs in mud and musk turtles. And
Yeah.
if that, because it's so dramatic that it's like within a
Right.
lifetime,
Yeah.
it's the whole skull is restructured.
Yeah.
So is there some level of, it kind of goes against the high school biology notion of change over generation. It's change within a generation to a certain extent.
Yeah. Well, and Australian side-neck turtles, the genus Emudura shows that megacephaly as well, and Elsea does. A bunch of them do. So I think there's the kind of almost pathological megacephaly, and that as I'm sure I mean, your bones are very, very fluid, plastic,
Right.
or organs in your body, right? Your bones are changing all the time, and they change with respect to stress, right? So you can whatever, if you've got a habit of cracking your knuckles all the time or something, that'll that puts certain stress points on your fingers, and it changes the structure of your bones. And your bones are actually a very responsive organ system.
Right.
And so my understanding is that some of that megacephaly has to do with the prey that they eat. If they're crunching mollusks, clams, crabs all the time, that puts different stresses and strains on the skull bones and the muscles. As the muscles hypertrophy, a lot of those muscles, like your temporalis muscles, attach on the outside of your skull, and if those expand, then they kind of pull on those bones, and that restructures your skull a bit. So there's that, and that's in many ways just sort of environmental plasticity or pathologies, I think. The more interesting one that I think is a really interesting area to learn about is things like in the genus Graptomes, the map turtles. There's there's two major clades in the genus Graptomes, and in one of the clades there's the narrow-headed and the broad-headed ones.
Yeah.
Right? So the broad-headed ones are things like pulchra and gibbon's eye, and they've got, the females have these really big heads, and the females sort of specialize on molluscivory, they eat mollusks. The males have much more normal-sized heads, and they tend to feed primarily on aquatic insects. How much of that is genetic, and how much of that is just environmental patterning that has to do with once you start eating mollusks, then you keep eating mollusks. The females also get bigger. Maybe they get to a point where they can start eating mollusks, then that hypertrophies their heads, whereas the males never get to that point. Or is there a genetic component? Usually in cases like that, some of it's genetic, some of it's environmental. And understanding those genes, I think, would be really interesting.
Yeah, what's controlling that?
What's controlling that, and do we see those same genes controlling megacephaly in other lineages where we see large-headedness in musk turtles or, in chelids or whatever.
We had Peter Lindemann on in one of the past
Yeah.
map turtles, and
Yeah.
he did something. I'm sure you've seen the paper where he looked sort of at the change of the characters over time and used some method independent contrast to look at kind of correct for phylogenetic
Right. Yes. Yep.
motion. But
Yep.
that's the next level, is figuring out where in the genome
Right.
these
That's
things
right.
coming
Yeah, that's
from,
right. And is it, I mean, that megacephaly tends to be kind of phylogenetically conserved. Some map turtles do it, Sternopterus does it, Endura does it. That might imply that, they're using the same genes, at least within map turtles, within graptomes. But is it the same genes as are being used over, musk turtles, where they haven't shared a common ancestor in 60 million years? Those are really interesting questions.
Where is the divide between phenotypic plasticity and genetics
and what fraction of that variation is due to one versus the other? And those are kind of classic questions in quantitative genetics that we can now get at in a very mechanistic way. Whereas we never could before. And that's part of the reason why, just as a project, we generated a decent reference genome for every family of turtles. And we just wanted that to be available as a resource to people, to be able to use that as at least a beginning platform to be able to do those kinds of analyses.
All right. Well, so we there's a few more just quick ones. I'm sort of curious what the most challenging project, and if the most challenging project you've ever done with turtles has also been the most rewarding. Those two things linked.
What's the most challenging project I've ever done with turtles? That's a good question. I would say the most challenging project I've ever done with turtles that I've finished. Let's go there, because I'm in the middle of one that's also horribly challenging. Was the paper that we just published a year ago, the Thompson et al. Paper on the phylogeny of turtles. And there we got, I don't remember, 85% of the species of turtles, of living turtles and tortoises on Earth, and built a big global phylogeny of them. And that project was challenging, not because the analysis was hard, not because of any of those technical things. It was challenging because it was so hard to get all the permits to be able to move those tissues across international borders and boundaries. The TSA, Turtle Survival Alliance, a lot of individuals there were really, really helpful. It was a great, collaboration. A lot of zoos chipped in stuff. But a lot of it too was just trying to get samples from other countries, and so many turtles now are protected under CITES, the Convention on the International Trade of Endangered Species. They're red listed on the IUCN. I think those are really important and really good moves for turtle conservation. But trying to then get all the permits in place across, probably 100 countries, it just was a nightmare. It was just really, really challenging, and lots of people helping us. So I'd say that was probably the most challenging single project. It took forever. Again, not to do the molecular work, but to get the permits. And we had to finally walk away from a bunch of things. We had to walk away from species that, I've held in my hands in the country of origin. I've caught them and collected them, but we could not get permission to move those samples across the border. I would also say that project was one of the most satisfying ones, just because of the scope and completeness of it, and a very, very, very satisfying project. So there you go.
Yeah, I'm always interested in sort of the driver of a lot. I mean, it takes a lot of effort, which
Mhm.
is like, and that's something for a lot of scientists. I think there's sort of this drive. Well, let's do the most cutting edge because that'll be the most rewarding and produce the most knowledge. So it's not necessarily a given correlation. So I was sort of curious.
No, it's not.
Yeah.
And it's definitely not. But you hope there's at least some correlation,
Right.
because
Yeah.
if
Yeah,
not,
you do.
you're just killing yourself for no good reason then,
Right.
right? And that doesn't make a lot of sense. Your time and effort and energy is better spent
Yeah.
than on assuming that projects that are satisfying to you are also projects that are sort of important for the discipline.
Right.
So that would be the hope.
So I'm assuming too, you're mostly in the lab, but I know you've done field work,
Mhm.
and I'm curious what the most interesting experience. It could be in the lab, maybe that's where it was, but doing turtle work, the most interesting experience you've had, something unexpected or interesting to you.
Oh, I mean, I think for me, so I did two sabbaticals in Australia, in Arthur George's lab, and I can't remember if you've interviewed Arthur or if you're going to, but
10 days.
in 10 days, yeah. So Arthur and I, as I was telling you earlier, we were born 2 days apart, and completely independently, both became turtle nuts, and got to know each other. Only I never met him. I went to Canberra and did my first sabbatical there, and there the main project the first time I was there. So you have to remember, when I went to Australia to work on the genus Hemidura, I had never thrown a turtle trap in the water before. I mean, that's more or less true. I mean, I was really new to the world of turtles. I had done a little tiny bit of work, and our goal was to trap Hemidura in every permanently flowing river from their southernmost extent, which is right around Sydney in New South Wales, all the way up the east coast, all the way around the Cape, all the way back around the Gulf of Carpentaria, over to the Kimberley Range in Western Australia, as well as the whole internal drainages of the Lake Erie drainage, the Cooper Creek drainage, and including the Murray-Darling, which is the biggest river system in Australia, and we did that. And I'd never been to Australia. We had an old beater, 25-year-old little Subaru that I bought from Scott Thompson, and it's just and it was just an adventure like you can't believe.
Oh, I'm sure.
There's figuring out how to trap turtles when there's saltwater crocodiles in the water. It was, being places where you were, 800 kilometers from the nearest paved road, and you're out there. I was out there with, my son, who was, and he did kindergarten in Canberra as well as eighth grade. But, out from, putting little, ornaments on eucalyptus trees Christmas Eve, and then, giving our son a present, because we're out camping in the middle of nowhere. Just it just from soup to nuts. It was just amazing, and getting to know Australia, getting to know, a lot of backcountry places, in New South Wales, Queensland, the Northern Territory, just sort of across that whole big chunk of the continent. Doing it all just by ourselves. It was really fun. It was really just really, really, really exciting. And getting to know the Australians was really great. I mean, there's other field work I've done in East Africa and Papua New Guinea and all across the US and Mexico and California that, has resulted probably in more papers or more that kind of output. But that experience was a real high point.
That's awesome.
Real high point.
And that's the stuff you can't, I mean, the data is great, but there's so much more.
It's part of, hopefully maturing as a scientist and or at least having a good time.
Right.
And meeting great people, and just a million great stories of some of them scary, some of them many just wonderful, but yeah, just really good.
Yeah.
Yep.
All right. Well, we'll just, our last question we ask everyone sort of as we started. If you have one piece of advice for someone looking to make turtle work a career, what would that be?
Turtle work.
Research,
Not research,
yeah,
but
anything. I mean,
anything.
yeah, related to turtles. Turtles is part or an entire career.
Yeah.
Yeah.
I would say, look seriously at yourself and ask yourself, why turtles? Why turtles for you? Okay. And build off of that. So, and if you just love them for whatever reason, that's one thing. If the fact that 2/3 of the species are tanking and you really care about conservation, that's a very different thing. If if you're a hobbyist and you really would like to be able to breed turtles, either because it would take pressure off natural populations for the herpetoculturist community, or just because you're interested, that's a different thing. But look deeply at yourself and understand why you care about them so much, and try and put that into a broader framework. And I think if you can do that, it will at least tell you, should I be, a person who's working hard to figure out how to breed them for conservation, or for the trade, or is it because I just find their ecology just so fascinating? Well, that pushes you in a different direction. You might work for an NGO like the Nature Conservancy. You might work for the National Park Service. You might be an academic like me. But I think that's what I would tell people is to try to understand that.
Yeah.
And then work with it. I think the other thing is that I would say is, people always say, well, as long as you're passionate, just follow the passion.
Yeah.
That's kind of true.
But it's so broad. Yeah.
There's a limited number of opportunities. So I think the other thing is, be reasonable, right? If there are people who, probably the best example is birds and the birding community, right? I mean, there's people who are, the most insanely, I'm a pretty good birder and I like birding a lot. There are people who are so good, it is just unbelievable how good they are. And, they're total weekend warriors. They do it, they've got a day job that pays the rent, and then they're birders, on the weekends, and they get immense satisfaction out of it. So it's not like you have to make a living at it, right? And you can make huge contributions. I think the TSA is a great example of that, of a collection of people who, are from all walks of life, zoos, academia, NGOs, as well as, it's kind of a lousy word, but hobbyists, who are deeply knowledgeable, really know their stuff, and have made major contributions to our knowledge of ecology, morphology, natural history, conservation. But they have a different day job, and I think any of those work, and you shouldn't be, I think, don't set yourself up for failure. Don't say, if I can't be a professional, herpetologist who studies turtles and that's all I do, then I'm going to consider myself a failure. That's the wrong attitude, and I think the right attitude is, find a way that works within, you got to pay the rent. You've got to be a responsible human being in other ways. And then within that, how can you make contributions in what areas that that you find personally satisfying?
I think that's really good advice, and all of us will take it for sure as best we can, and
Very
hopefully
good.
listeners can. I got to ask one more thing because you did sort of mention this, and I asked you this in Spokane last year, and I thought your response was really interesting. Why turtles? Why should people care about turtles? Why is it necessary?
I don't remember what I said in Spokane.
Well,
so,
yeah, I mean, whatever the answer.
why? Well, I mean, so I mean, I think each individual sort of answers that question themselves. If you
Yeah.
say, well, why are you interested in turtles, or why am I interested in turtles? Those might, for me, maybe it was those snapping turtles when I was a kid,
Yeah.
in my yard in Connecticut. I mean, I think in a sort of general sense, why should people study turtles, and why should people care about turtles? I mean, they are every lineage is unique. Every species is unique, right? If it wasn't, we wouldn't recognize it as a species. So by definition, every lineage is unique. Turtles are, they're bristling with so many unique features that have never evolved any other time.
Yeah.
I mean, the quintessential turtle character is, putting your shoulder girdle inside your rib cage, right? I mean, just ours floats on the outside of our rib
Right.
cage, right? Like any normal, well-constructed animal. Just imagine that your arms originated inside your rib cage. You'd be walking around like this all the time
Yeah.
because there's no room to maneuver. I mean, turtles are just they're so bizarre that I think that sort of just makes them a target for study to try to understand how in the world this kind of crazy evolutionary novelty could have ever existed. And you combine that with the fact that, we just ruthlessly kill turtles, and I mean, from a conservation point of view, I've always felt turtles in many ways are a really interesting, unique opportunity for me as a conservation biologist, because we know exactly why 2/3 of the world's turtles are going extinct, right? And it's because we kill them.
Yeah.
And that's something we can fix, right? That's, we don't have to kill turtles, and yet we do. And, for the most part, it doesn't appear to be pollution. It doesn't, they're very flexible, I mean, not every species, some are very fussy, but an awful lot of them are very flexible, relatively generalist species that, they can take a lot of heat. And so as a conservation problem in conservation, it's as much about awareness and human perception as it is anything else. And that's something you can actually sink your teeth into, and you can actually do something about. And so I think that's another reason to study them.
Yeah, that's just interesting to ask everyone why, because that's
Mhm.
sort of the main. I think a lot of us are still trying to figure it out.
Yeah.
I don't know exactly.
Yeah,
It's just
yeah.
this,
And sometimes it's just some kind of,
really.
Little kind of obsession that you have.
Yeah.
And as a colleague of mine said to me one time, you can spend your life trying to figure out why you're so fascinated by something. On the other hand, if it works for you, just go with it.
Do it. Yeah. I think that's a good way to finish it off.
Yep.
We've gone with time, but we also, we don't have to do this if you need to go, but we do like to do just a quick turtle trivia round at the end of everything. It's just a way to bring in weird random turtle
Okay.
facts. So
Okay.
I don't know if you've got a few turtle questions you want to toss up to us and we can try to answer them.
So this is me asking you guys.
Yeah,
Turtle trivia.
we can give
Okay.
you some if you want.
Well, let me think.
I like to tell people beforehand, but the past 10 people I've invited, and I've forgotten to do it. It's just on the spot.
It's on the spot. Okay. Let me think. So, a few questions for you guys.
Yeah, anything.
What turtle has the smallest geographic range in the world?
Oh, that's a good one. I know the largest.
And what's the largest?
The largest is the scorpion mud turtle. Oh, we're counting. Okay.
Are we?
Yeah. Well, I'm
All
just
right.
conditioned not
Okay.
to.
All right. So if Scorpioides got split up, though, then that would kill that. But what's the smallest?
The smallest range.
I'm not sure.
And Wyatt, you guys want to pitch in here?
I mean, I want to say
It's probably red-eared sliders. Right?
The smallest range.
I
mean, I don't know. I'm seeing if
I-
you guys can kick in on it. What would you think?
the Kwahulian box turtle comes to mind, but
Which one?
Yeah, that's
Kwahulian box turtle.
Yeah. I wonder, and I mean, it's just there in Quashcienagus. That's probably a good one. I was going to go with Graptomys pearlensis,
Okay.
which is just in the kind of lower Pearl River, which is a pretty small drainage. But I think Coela is a better one. Are there any? Now, of course, there's a few species where we really don't know their native range. There's a few Southeast Asian species of Moranis and stuff that we don't know, or Quoras.
geometric tortoises could.
They've got a really small range too, and I don't know how big the ranges of the Pyxis species are, but they're pretty small as well. But I'll bet you Coela, Terracotta Coela. I think that's a really good guess.
That's good.
Okay, let me think if I can come up
All
with
right.
another one.
It's always tough on the spot.
Yes. I'll keep thinking. You got one for me?
I'm thinking of something. That's the Ken, Wyatt. You guys got a question on hand?
I got a really easy one, but I thought it was fun because we talked about it a little bit earlier. What is the only amidid species outside of North America?
There's two, right?
Oh shit! Okay, outside of. You're right. I totally
Right.
disregarded.
There's Orbicularis and Trinacris.
Yeah.
Yeah, you got it.
Yeah.
Very good.
Thank you. Okay, let me think. What turtle has the northernmost range distribution?
Well, sea turtles.
Excluding sea turtles.
Okay. This one we are excluding sea
Well,
turtles.
excluding sea turtles.
That's another. Well, okay, so sliders technically.
Yeah.
I did some work with it. They're 55 degrees north latitude in Russia. They've been reported, but they're not. It's
But
not
they're not
viable.
native. Okay.
Yeah, and they died. So
So native.
Yeah, okay, native,
I'll
native.
read, yeah, native. What would you say?
Palladiscus sinensis is a pretty far north range, doesn't it?
Which one?
Palladiscus sinensis.
Yeah,
European.
but I think that Horsefield eye, the Russian tortoise, I think probably gets the furthest north.
Yeah, that makes sense.
And I'm pretty sure
Almost.
that's right. We'd have to check. I don't know if you guys know. You probably do. Our turtle taxonomy working group, checklist of turtles of the world.
We had Anders on.
Okay, yes.
Yeah.
And now we've got really good, accurate range maps, and as well as body sizes and stuff of every species on Earth. So, I should know that since I've been an author on that since the very beginning. But I think it's Horsefield eye. I think that's right.
It's close between that whole.
Yeah, right. But
No, it's
but
good.
I'm pretty sure they would be the most northern.
Huh, cool.
Yeah.
All right.
That's great.
Okay.
Well, that was good. And I think we'll end it there. For everyone listening, this is episode 41. Dr. Brad Shaffer, thank you again for coming on.
And
It
thank
was-
you guys. This was a lot of fun, and really a lot of fun. And I'm gonna have to go back and look at the other 40.
Yeah.
So-
Yeah. I'll send you a link. But yeah,
Okay.
it's been an honor, and just for all of us, I think, just growing
Yeah.
up reading all of your stuff. It's really
Well, thank
yeah,
you. Thank you very, very much. Really appreciate it.
cool. We'll
Okay.
see you in the
Keep
next
it up,
one.
you guys.